Welcome to Module 8 of the course on genomic selection. And in this module, our last module, we're going to talk about other considerations that can affect genomic selection. Our topics are shown here: trait heritability, genotype-environment interactions, ways to optimize the training population, combining GS and marker-assisted selection, effect of genomic selection on diversity, parent selection, and the end products of genomic selection. So one thing we have learned about genomic selection is that if your heritability of your phenotypes from your training population is low, then your genomic selection, the accuracy of genomic selection will also be low. And that's just shown here in this data from one of my studies in wheat with 6 traits. And what we see here is very distinct, that if we have high heritability, we have high accuracy. And as we have lower heritability, we have lower accuracy. And almost every study shows this exactly the same thing. It certainly shows that good phenotypic data, quantity and quality, will lead to high heritability and therefore lead to higher accuracy of genomic selection. Another factor on genomic selection is dealing with the genotype-environment interactions. The G/E interactions arises from several sources. One is the low correlation of genetic values between environments. Now, in true genotype-environment interaction, genes really have different effects in different environments. But G/E really is just a statistical thing, and some of the times your G/E simply arises from experimental error. In other words, your phenotypes are not that accurate, and therefore you don't have accurate estimates of genetic values, and therefore they can't possibly correlate to genetic values from other environments. An important point here, though, is that your phenotypes from your training population must be able to predict the value of genetic values that will occur in future environments. So basically the G/E between past and future environments must be low. And we just show this schematically here. You have a training population, what's called set A of lines, and these have all been phenotyped in set 1 of environments. You're going to use that data to build your training population, build your GS model. All that phenotypic data comes from past environments. Now let's say you're going to take that same set of lines and you're going to phenotype them in set 2 of environments. The question is, now can a model built on data from set 1 of environments predict the phenotypes that will occur in set 2 of environments? You're using data from the past to build your model, and you're hoping that predicts performance in future environments. And this is really the crux of plant breeding. And we do this with our phenotypic data as well as genomic selection. Can data from the past predict performance in the future? Well, this shows some results of studies we've done in my program. We're looking at the phenotypic correlation between data collected in 2 different environments. That's along my x-axis. And then we use data from one set of environments, one environment, to build a GS model and see if it can predict the phenotypes in the second set of environments. In other words, the GS accuracy between environments. And what you see here is when we have low phenotypic correlation between the environments used to build the model and the environments used to validate it. When we have low phenotypic correlations, we also have low GS accuracy between them. In other words, the phenotypes in these 2 environments don't predict each other. And so the phenotypic data from one environment used to build a GS model doesn't produce a model that can predict the phenotypes in the other environment either, which all kind of makes sense. And then when you have these low correlations, that usually means you have high G/E. And here's the other end. You have low G/E. The correlation of phenotypes in one environment and the other are quite high. This oftentimes happens with our trait like height and even maturity. And when that occurs, we have high GS accuracy. So again, it comes to a point past data, phenotypic data, must be able to predict the future if you want your GS models to be able to predict performance in different environments. Now there's also ways you can try to optimize your training population. The rule of thumb is more markers and more lines, the better off you are. But that's not always true. So you like to look at your data and see if can you reduce the noise in the data and the redundancy. In all data sets, doesn't matter what it is, your data can be broken down into two parts: pattern, which is useful for predicting the future, and noise, which is just experimental error, any type of data that does not lead to improved predictions. And so basically your pattern is data that leads predictions, and the noise is really non-predictive components of the data. So when you're looking at your training population, you can consider using subsets of markers instead of all markers. You can consider using subsets of line instead of all lines. One thing is, instead of using all markers, you can try to choose markers that provide equal coverage of the genome. In other words, reduce the number of markers, and so you have less redundant markers. And this will allow for easier, faster analysis and probably less biased. For example, I know in wheat, we get thousands of markers sometimes from chromosome 2b, and very few markers from some of our other regions of the genome. So we don't want to use all these markers from that one chromosome. We want to reduce that because most of them are highly redundant to one another. So we filter them out so we have less redundancy in our markers. The other thing you can look at is using only markers that are significantly associated with a trait. So in this first step, you're reducing redundancy by throwing out markers that are all from the same region of the genome. In the second part here, you're using only significant markers. These are the ones most likely associated with genes controlling your trait. And if you use those, you're probably just throwing out the noisy markers, the ones that are not associated with the trait, which really can't possibly begin to add to predictive value. You can also look using subsets of lines. Lines with low G by E. These are lines that are the most predictive. These are lines that have more pattern to them and less noise. So if you throw out the ones that have high G by E, that's a lot of noise, throw them out, and you probably get a better prediction model. And also you can look at your population. You look at the genetic structure of your population, and you look at clusters of lines. You may want to subsample instead of testing, let's say genotype 500 lines, and you find out that there are two distinct clusters. And you don't really need to take all lines from both clusters. You can sample lines from within each cluster to represent the overall structure and just phenotype those. And that is basically looking at ways to reduce the redundancy in your training population. And again, this can make you more efficient. So let's look at what happens when we try to subset markers using only markers that are significant versus using all markers to build our models. So down here, we're looking at different traits and wheat traits, again from my program. Here's our accuracy. We build our model using all the markers that were scored on the lines in our training population. And on the y-axis is the accuracy we get if we use only the markers that are significant at a p-value of 0.05 when we do an association analysis. So using all markers, our average GS accuracy is 0.46. If we use only the significant markers, our accuracy jumps up down to an average of 0.74. So that's a great increase in our accuracy. And in this case, we're using on average just 5.8% of all the markers from that total marker data set. So now with just very few markers, we actually have improved our GS accuracy because we're picking only the markers that are really associated with pattern in our data set and predictive, versus all markers when the majority of those are really not very predictive at all. This also helps us with our accuracy of genomic selection between environments, using data from one set of one environment to build a model and see if it can predict the phenotype in the second environment. So when we look at our accuracy between environments using all markers that were scored in our training population, the average accuracy is 0.24. Okay, not too good. And now when we use only significant markers, our accuracy between environments is now 0.34. Still not that great, but it's better than it was. So we have improved our accuracy of predictions between environments, which again is really one of the most important aspects of using genomic selection and in plant breeding. We'll continue with this topic in the second part of Module 8.